Welcome back to Chapter 15, Mutations. We're going to continue on and talk about the way that mutations can occur, and then start to talk about some of the diseases that are known to arise from some of these different mutations. I want to look at some transposable elements, as well as a little bit about the Ames test and get you ready for doing the homework. So mutations, and I kind of broke in the middle here because of time-wise this made sense as a break. But mutations can be caused by chemicals that are, or other things that are mutagens. This is the symbol for it. We notice we don't have that in the lab at all. The thing that we normally would use to stain DNA to look at it in the gels is a known mutagen. So we use other stains instead, and they're just not as effective, but they also don't mutate your DNA. So we can deal with that. We're always exposed to a whole bunch of plethora means a lot of different kinds of mutations. I think the most common one that you're exposed to that you don't think about is ultraviolet light. That's coming from the sun. The first mutagens were actually identified in mustard gas in fruit flies in the 1940s. I want you to think about that it hasn't been 100 years since we've known that we can damage ourselves with chemicals on a fundamental code level. So here I have one of the things that the mutation can do is substitute either a pyrimidine or a purine with something that is like a base, a nucleic base, but is not. So this chemical here, 5-bromouracil, it acts like thymine. So analog means it's similar but not exactly the same. There are other forms of chemicals that act as a cytosine analog. The body thinks that it's a cytosine, but it's not. And that means it can bind to DNA in the way that your thymine or cytosine would without actually holding on to any of the information. When we divide this again, this is not going to be the same piece of information. What can happen is my wild type, A to T, I accidentally insert my bromouracil, 5-bromouracil, and as that repeats, that thing can change and bond to a different piece of information. My original was A to T, but now this 5-bromouracil will now bind to a guanine. It becomes a cytosine analog, and now I've completely reversed my code. It codes for something completely different. This is a point mutation. Maybe it has a huge effect. Maybe it doesn't have an effect at all. Alkylating agents can donate an alkyl group, a methyl, or two methyls to amino or keto groups, and they alter the affinity or the likelihood that the base pair will actually bind together. They can result in transition mutations. This is what mustard gas does. And you know mustard gas is super bad for you. It's deadly. What it does is it suffocates you, but it also interferes with your DNA synthesis and changes the code and binds to it so it can't separate from itself. Intercalating agents are things like ethidium bromide. This is what, in my lab, this is what we used for looking at the bands in gel electrophoresis. This chemical that causes it to glow under a black light is called ethidium bromide. If you get it on your skin, what it does is it inserts itself. This is the ethidium bromide. It inserts itself in between one strand of DNA and the anti-parallel but complementary strand on the other side, and it pushes it and causes a distortion in the shape of it. That structurally causes it to be less likely to hold together, but it also causes it to tear and then insert more mistakes. The reason we don't use it in lab is because it messes with your DNA. It's never a good idea. Adduct-forming agents causing chemicals that bind to the DNA. They adduct it, they move it away, and that alters the conformation and so the actual shape that it's supposed to do. It interferes with repair and replication processes in the DNA. Acetaldehyde is something that is found in cigarette smoke, and this is actually how it acts. It causes your DNA to be more likely to break and changing its conformational change means the actual shape that it's supposed to take. I want to put this here, HCAs. I didn't make it red. I'm not going to have like a list of chemicals on the exam, but I still want to talk about it. We know that these are tied to cancer. We know they are cancer-causing agents. When you cook your food and you blacken it, not with spice, but you actually blacken it in oil, or you cook it so it has this black, and you char it. Char is a better word. And you char it, that actually creates chemicals that are damaging to your DNA. They've been tied to breast, colon, and stomach cancers. You can avoid them by not charring your food. I think this is part of the argument of using different kinds of oils when you cook. Olive oil smokes at a very low temperature. So if you're frying something in hot oil, olive oil may not be the best choice because it's more likely to convert into cancer-causing agents. You may want to use something like a corn oil in that case. Olive oil is better for colder dishes because of these chemicals. UV light, you've heard your whole life that the sun is damaging to your skin. The UV light damages your skin by actually causing your DNA structure to not be as stable and to fracture and be a little bit wobbly. It breaks some of the protein structures in your proteins as well. Some organisms, not you or me, can use photoactive repair to reverse this, but this is a bacterial thing. Ionizing radiation is amazing. It comes from outer space, things like X-rays and gamma rays and cosmic rays. When they hit you, they cause you to mutate and gain superpowers. Cancer. No, sorry, I meant it makes you have cancer. In all the comic books, they are amazing things, and it's changed over the years as the different comic book characters realize, as the authors realize that these things are getting, obviously, those are cancer-causing agents and not causing mutations that are favorable, very likely, but maybe. Even more likely to get leukemia from these things. The chances that you're exposed to too many cosmic rays are pretty low if you're staying on planet Earth. Gamma rays also pretty low, but X-rays, we do monitor our X-ray exposure. If you work with X-rays, you wear my husband works with X-rays in a machine. He wears a tag that monitors his X-ray exposure per year, make sure he's never going over. These are things that ionizing, it means that it turns some of your molecules into ions, and you can maybe repair this with some good diet, but mostly it's good to just avoid these situations. Too much. A little bit is actually not enough to damage you. Of the things that are actually affecting you, radon gas is one of the bigger concerns. If you have a basement dug down into the bedrock, we monitor you for radon gas. That is something that can just be produced by the environment around you, highly tied to causing DNA damage, soil radioactivity, isotopes in your body, eating things that are exposing you to this, radioactive pharmaceuticals. I think this is not a problem so much anymore, but if you think about some of the paints and chemicals that have been used in the last 100 years, absolutely, exposing you to DNA damage. Nuclear weapon fallout, again, not a huge deal, hopefully, to us right now, but is actually at a higher level in our environment than it was 200 years ago, before nuclear power was discovered. This here, these are, your normal exposure to these is very low, and we monitor them to make sure because they cause damage in your gametes as well as in your somatic cells. Most genes that we talk about and most diseases that we talk about are polygenic, meaning more than one gene is involved in that. There are some very well-known monogenic or single gene problem diseases, things like achondroplasia, which is a form of dwarfism, Marfan syndrome, which I feel like we talked about in a couple lectures. This is familial high cholesterol, cystic fibrosis, Huntington disease. These are all caused by a single change, one single point mutation that has caused a cascade of problems that lead to a protein malfunction. These can be heritable, and this is what we did with our Punnett squares. We traced them through family lines. You do have some repair systems. Bacteria have repair systems that actually counteract spontaneous and induced mutation. So whether they just happen or we've actually caused them by a chemical, this double checking maintains the original code. Your DNA polymerase proofreads, it backtracks, it makes sure that it made the right match, and then DNA polymerase 1 can actually replace incorrectly inserted nucleotides and do some mismatch repair. How does it know which side is right? Like if I make a mistake and I've gone like I had A, T, T, C, and my DNA mechanism put T, C, let's make it a G, A, G. How do I know that that was the mistake and this was right? When you use your DNA and it goes through the processes of actual function in a functioning cell, those pieces of DNA get methyl groups attached to them. They become methylated. When it's separated during DNA replication, the new strand is not yet methylated. So as you create those new strands and you have the parent and the daughter strands, the parent is actually identified as the parent by having methyl groups attached. It's a little bit older. And so the replication mechanism can recognize who was maybe not right, but who is the original copy. If the DNA polymerase actually skips over a break in DNA, you can actually, with some of the enzymes in your body, repair a broken piece. The broken piece is lysed, it's cut out, and then repaired in with a series of proteins. The REC-A protein actually directs recombination exchange. It's looking for the undamaged strand and the newly damaged strand, and then tries to match what was the appropriate nucleotide that was supposed to go in there, and then that gap can be repaired. We can excise out pieces that are not correct. DNA polymerase inserts the complementary match after excision of the one that is wrong. DNA pol 1 inserts the correct match to the original parent strands, and then DNA ligases seal the space in between. They reform those phosphodiester bonds. This uses a base excision repair and a nucleotide excision repair, depending on if you pick a base pair, A to T, or just repairing a T or an A, or a G or a C. I don't want to give you the impression it matters what letter. The Ames test. I have this listed in a little video, I think, in your homework to go through this. I don't think you need to analyze this as much as the textbook does, but the textbook has an entire chapter on this. I think that the guide that was published in the homework is an excellent overview of this. I think I made it so it activates. The Ames test uses a bacteria that is autotrophic, it's self-feeding, to test if a chemical is a mutagen. What is going to happen, here, I'll read these words to you. So if we're going to test a substance in a extract of bacteria, Salmonella, with some extract of rat liver, and I'll explain why. If the test says it's positive, it may be that that particular substance is actually a mutagen. We're going to use a couple different strains of bacteria. They're Salmonella, the specific species. They're auxotrophic mutants. They cannot produce the amino acid histidine. I grow them on a plate with minimal histidine. As they grow, the first couple bacteria, as they divide, use up all the histidines inside. And if anyone survives, that means they had a mutation that allowed them to, over time, make histidine. So they revert it back to their original ancient form. I want to put a little bit of histidine on this plate because I want to give several generations the potential to mutate. They are exposed to a chemical that I think might be a mutagen, so I'm encouraging them to mutate if possible. If they mutate, now I can say that this chemical is actually a mutagen. Now there's always a chance that the mutations are spontaneous, so I run a second plate where I don't expose them to the mutagen, and I see if any of them survive without exposure to the mutagen. This then determines my sort of natural background reversions. So how many mutations are happening spontaneously versus how many mutations are induced by the chemical. When I do this, and this substance is determined to be a mutagen, I might just run it here in this plate all by itself and expose it into the mutagen by itself in a bacterium. Or I might make it grow in a liver extract, so rat liver extract. What that does is allow some of the metabolic processes to occur that would occur in a normal living organism. So I can see if it may not be the mutagen itself directly, but some byproduct of the mutagen that is actually causing the mutations. So I can see, is it this chemical, or could it be this chemical inside my body going through the changes that my body is asking it to do that is actually causing the mutation to happen? And both of those are of a concern because we're worried about having this process be a mutagen in living human beings, not in cells on a plate. That's not our concern. Transposable elements are those jumping genes. They move between chromosomes. They're very common. I've got an example here of transposons. I have these little jumping genes, these little things here, these little pigments are representing where genes have jumped from place to place. They're very common. We think that they have a major role in making our immune system work so that things move around and allow us from chromosome to chromosome and allow us to respond adaptively to diseases as they infect our bodies in a way that is beyond just sort of randomly generating our immune response proteins. They also are characterizing some diseases that we're seeing where genes end up inserted in places where they're not supposed to be, either interrupting normal function or in silencing genes that are supposed to be expressed. And we have a lecture in maybe another week or two after the exam, where I'm going to talk a little bit about epigenetics. We can have an additional above the genes control of expression that we haven't really gotten to the heart of yet, that DNA transposons can play a role in. There are two kinds. There are transposons that move around without the help of RNA. It has this is a strand of DNA. This is the transposon, the piece that is going to jump around. The DNA, when this is the place that's going to jump around. The transposon includes a code for a protein called transposase. It's an enzyme that causes transposing. This enzyme itself cleaves this section at these regions at the end, the ITR regions, and allow it to then jump into another segment of DNA. This is anywhere from 9 to 40 base pairs. They're really, really short little jumping pieces. Retrotransposons, retro means going backwards, move from a location to location with the help of RNA. They have more of a copy-paste mechanism. They make this enzyme transcriptase makes mRNA copies that gets translated into reverse transcriptase, which runs transcription backwards, and a protein called integrase. These are both enzymes. A's and A's on the end tell you they are enzymes. They reverse transcriptase takes the mRNA copies and writes them back into DNA, so cDNA, complementary DNA, and then inserts them back into the DNA strand. I want to point out some fun things here that we've used in chapter 20 at the review, and your body is also using here. In order to make this region fit well into the DNA, the mRNA has poly A tails. The DNA, where it gets inserts, excuse me, has a poly T tail so that they bind really well together. And we take advantage of this when we're doing some DNA technologies later. So I guarantee this was a little bit listy. some ideas that we need to come back to. I really want you to integrate the idea of DNA gone wrong in our process of DNA replication, but also the consequences of what happens when we have an mRNA converted into a protein translated, and that protein is now not functioning because it has the wrong code, whether that was a base pair mistake or if it was an entire piece that got shoved in from somewhere else. That affects the phenotype of the individual that is actually using that DNA. I have 2 more questions for you, nice and easy today. What's the potential effect of a transposon in the genome? I covered it in 2 different places. All of them. So gene inactivation, putting it in and putting it in a place that kind of ruins a gene, changing in the way we regulate it by epigenetics, we'll come back to that, but outside control that regulates a gene that had nothing to do with our transposon, altering where we can put genes together and put them apart. And it also, even though I did not highlight it, it might actually involve messing up the exons. And now if I insert it in between where exons would be, I may actually shuffle my exons around. And this is incredibly important in your immune system, as well as some other things. Finally, here's a graph. It's the same graph it was in my lecture. It shows the effect of X-rays on the number of mutations induced. You may need to make it bigger, but it's the same one in the text too. What is it saying? You ready? Any exposure to X-rays can cause mutations. There's a linear relationship here. So this is a lot. This is a big amount of X-rays, but you should limit your X-ray exposure as much as possible. That's why if you work in a place where you give X-rays, you do the thing where you line everybody for X-rays and then run out of the room. You know, you put the animals in the tube or something, and you don't stay there and hold them generally, because you're, especially over time, the more often you expose yourself, the more DNA damage you are doing. Well, with that, I guess that's the end. I feel like go back to here real fast. Little bit of review of all of the things that we've talked about for mutation. A lot of this we have already covered. I truly did leave some of this for the homework for you to puzzle out, because I feel like we've covered it so many different ways. I think that you can, there's 2 things that I put notes-wise in the homework that I think you should prepare ahead of time. One of them is there's one of the questions, part B, only has 2 answers, but it's not clear from the way it's written. And another hint at solving some of the problems. Watch the little videos that come with it. There's 2 or 3 videos that are 5 minutes long as part of the homework that I think make everything move around and make it a little bit clearer. I'm going to go ahead and record 16. I'll see you in class this week. We're going to solve some crimes. All right. See you soon. Bye-bye.